confirmed on February 8th by peer-reviewed Virology Journal, Volume 16, publication date April 2020. So this is coming out next month, but I will show you. In fact, here is the study, and you can see published date is April 2020, and it's talking about the spike glycoprotein of the new coronavirus 2019 nCoV, which is basically the COVID-19, contains a furin-like cleavage site absent in coronavirus of the same clade. So this furin-like cleavage site is absent in the regular coronavirus. It is present in COVID-19. That's very, very important, and I will come back to that. There's an underlying implication here that it's very subtle, but the only possible reason for this modification is the creation of a biological warfare weapon. There are no other plausible explanations. Now that sounds a little far-fetched, I agree, but wait, you're going to understand. Because when you start understanding this furin cleavage component that we're talking about, then it starts to make a little bit of sense. But why would somebody do that? Well, or how can we prove that somebody actually did that? So we're going to talk about that. Now, this virus was actually created at the University of North Carolina in Chapel Hill, and they published the highlights of the creation in November 2015. The list of scholars associated with this Frankenstein of a virus, you'll see, named Zheng Li-Shi, who just happens to represent the key laboratory of special pathogens and biosafety, Wuhan Institute of Virology, Chinese Academy of Sciences, Wuhan, China. Of course, there were other scientists, but most of them representing the University of North Carolina, Chapel Hill location, but there were also representatives from the Bellinzona Institute of Microbiology from Zurich, Switzerland, as well as Harvard Medical School. All right. Now, Dr. Shi, she's a key figure here. In 2014, Dr. Shi was the recipient of a number of U.S. government grants, as well as grants from the National Basic Research Program of China, the Chinese Academy of Science, the National Natural Science Foundation of China, and from the Strategic Priority Research Program of Chinese Academy of Sciences to assist in funding research into coronaviruses. Now, this is, remember, 2015. All right. What's interesting is that the U.S. government paid Dr. Shi and the Chinese government to fund research into coronaviruses. Now, what did we get for this research that we paid, the taxpayer money went for? So this is what we got. Using the SARS-CoV genetic system, it goes into some specifics on the type of viruses that we're looking at, the backbone of the virus. The results indicate that group 2B viruses encoding the specific version that they were looking at in a wild-type backbone. This is SHC014 spike in a wild-type backbone, meaning the naturally occurring virus, can efficiently use multiple orthologs of the SARS receptor human angiotensin-converting enzyme ACE2. Now, I want to talk about this issue with the angiotensin-converting enzymes in a later video. It's not pertinent here, but it is pertinent when we start talking about some of the misinformation regarding treatments that's coming out. Goes on to say that both monoclonal antibody and vaccine approaches failed to neutralize and protect from infection with coronavirus using the novel spike protein. So this novel spike protein was essentially something that they introduced into the coronavirus, and it essentially was a morphological departure from the naturally occurring coronavirus using the backbone of the virus. The implications are very, very suspect. All right, so they took SARS, the virus that caused SARS in 2002, 2003, and that was carried by bats, making the rounds back in 2003, and reverse engineered the genetic coding itself inside the virus. And they took the backbone of the original wild-type virus, and that's when they started messing around with it. They then inserted additional proteins to enhance the efficacy of the virus inside the human lungs, specifically for the human aspect, making it extremely difficult to vaccinate against because the new virus has multiple proteins with which to attack. So basically what they did was they made a bug, a virus, into a super virus. What would be the purpose of doing such a thing? Well, it'll become self-evident here in a few moments. What's also interesting is that these coronaviruses that they took from the SARS component and then they manipulated them, these cells were originally obtained from Fort Detrick. You may remember that Fort Detrick was also where some of the other HIV testing had been done and research had been done. And my friend Garth Nicholson, who used to be the director of laboratory at MD Anderson, wrote a book called Project Daylily about his work with some of the stuff that happened with the special forces, some of the research that was done at Fort Detrick, some of the stuff that was done from in the HIV arena. And again, a lot of the stuff was done at Fort Detrick. So it's interesting that the correlation, this is like 20, 25 years ago. All right, so they say in the paper in 2015, remember they went back, this is going on from 2003 to 2015 now, they were looking at the SARS component from 2003, the coronavirus that caused SARS, and they were now manipulating that, and they published this manipulation study, but after they manipulated it, they published it in this 2015 paper, and they created this hybrid virus, which is far more deadly and can't be vaccinated against. It is resistant to vaccination because of certain morphological changes that were created to the wild-type backbone. The new DNA, genetically engineered virus, constitutes a gain in pathogenesis. Now, I want you to remember what gain in pathogenesis means, okay? Or gain of function is the term that they use in virology, or actually in research. So gain of function. Okay, here it is. That's a gain of function right there. They admit it. Gain of function basically means taking something and making it more potent, all right? So pathogenesis means the lethalness and the infectiousness of something. The pathogenesis is the cause of the actual problem, cause of the disease. And Dr. Shi learns how to perform gain of function operations within viruses, and then she returns to her post in Wuhan, China. So she learned this in University of North Carolina, Chapel Hill. They get the cells from Fort Detrick, and now she goes back to Wuhan, which is where the techniques that she learned about gain of function were then applied. Fast forward to November 2019, a breach of containment occurs at the Wuhan Institute of Virology, and patient 0 is detected somewhere between December 1st and December 12th. Now, we don't know about the details. We don't know who the patient 0 was, but apparently there was a breach and containment. Now, I'm just going to quickly go over this. Dr. Shi is one of the authors that published this paper in January 23rd, 2020, stating that it is of probable bat origin. The problem is she knows what it is and what the origin is because she was the one who created it. Okay, she helped to enhance it. This is an enhanced version of the hybrid virus that they helped develop at UNC with Fort Detrick support back in 2015. Dr. Shi knows this because she helped to build it. Now, what does gain of function mean? Again, as I mentioned, gain of function is basically that it made it more potent, it made it more virulent, it made it more deadly, it made it more resistant, it made it more effective, and made it into a superbug that nobody has basically seen before. That's what that means, okay? And it's also remember that this good doctor and her team inserted HIV and MERS proteins into the coronavirus to give it just a touch more kick. This is part of what they did with the coronavirus. So you're taking HIV and MERS, both causes of a lot of disease, a lot of death, a lot of mayhem, and they inserted proteins from HIV and MERS into the coronavirus. So they took proteins from one pathogen cause one disease, another pathogen cause another disease, and they put both of those into this hybrid virus that is now known as COVID-19.